Yeah, guys. Fast forward to molecular biochemistry. Today we'll be talking about the central dogma of molecular biochemistry. Central dogma of molecular biochemistry. All right. So now, if they ask you what is the central dogma of molecular biochemistry, how do you go about this question? Of course, the central dogma just have to do with protein being transcribed into RNA, then RNA being translated into protein. Sorry, DNA being transcribed to RNA, RNA being translated to protein, okay? So we're talking about this process and why it is referred to as what the central dogma of molecular biochemistry. So basically, the central dogma of biochemistry is a framework that explains the flow of genetic information. Of course, the RNA has a genetic information. It has to translate it into he has to give it to the DNA has the DNA the genetic information. It has to give it to the ribosome so as to process it and then form what functional proteins. Okay, so it shows the flow of information within a biological system. Okay, so it was first proposed by what Francis Crick in the year 1957. Okay, proposed in 1957 but published in 1958. So now, the central dogma states that genetic information flows from genetic information flows from DNA to RNA, now from RNA towards protein. So in more details, the transfer of information from nucleic acid, okay, to RNA or nucleic acid to protein may be possible. So, but transfer from what protein to protein or from protein to nucleic acid is what impossible. So information is actually transformed from what nucleic acid to protein, okay? From nucleic acid broken down towards RNA, from RNA to protein, okay? It can be information cannot be translated from protein to what and information cannot be transported from what nucleic acid to protein, okay? So that's what that statement means. So this is DNA. DNA can actually still be replicated, okay? So now breaking DNA down to RNA has to do with what transcription. Then turning RNA to protein has to do with what translation. Okay, so that's it, guys. Now, so that information here means the precise determination of sequence. You know, amino acid or proteins are actually in sequence of three. You have the AUG sequence, you have the you have the UAA sequence, and so on. Okay, so this information we are talking about has to do with the precise determination of the sequence, okay, either of the base in the nucleic acid or of the amino acid residues in the protein. Okay, so now the central dogma is a framework for understanding the transfer of sequence information between information carrying biopolymers, okay, in the most common or general case in what living organisms. So there are three major classes of such biopolymers, which include what DNA and RNA. So both of them are nucleic acid and also protein. Okay. So now there are what 3 times 3 conceivable direct transformation of what information that can occur between this. Okay. So since we have three biopolymers, there are about 9 ways in which these biopolymers can actually interact for the flow of information. I've seen this before, right? And basically, guys, that's the introduction to the central dogma of molecular biochemistry. Okay, a study of how there's a flow of genetic information within a biological system. Okay, so this I'd like to close it up and see you guys in the next tutorial. Bye for now.